So take it away, Noah. You got some background there.
Yeah, I apologize. Busy lab.
We also lost him, right?
Yeah. No, also you just, I don't know if you can hear us with the Zoom freeze. So Diana, you ready to go?
Sure, let me do that. Of course. So thank you all for inviting us to be here today. So the title of the work that we're going to be talking about is personalized ex vivo drug screening and genomics with AI machine learning for individualized cancer treatments. Oh, you know what? I don't have control of this.
Yeah. Well,
Am I back?
Yes,
yeah, you're back. There's
okay, good. I'm not
go.
allowed to have good internet. That is one of the rules of the world. But yes. So yeah, I'll just go to the next. Wait. Okay. So quick intros from us. Diana, do you want to go first?
Yes, absolutely. So my name is Diana Azzam, and I'm an assistant professor at Florida International University. My PhD was focused on ovarian cancer stem cells, and then my postdoc was focused on high throughput drug discovery. And so when I joined FIU as an independent faculty, my research interests include implementation of functional precision medicine in clinical trials to guide individualized treatments in advanced cancer patients, as well as a major focus of my lab is to understand the resistance of cancer stem cells and how they play a role in metastasis.
Thanks, Diana. And I'm Noah. So I have a couple roles. Right now I'm most accurately serving as founder and CTO of First Ascent Biomedical. My background is electrical engineering, computer science, and math. Did my PhD in electrical engineering. And during that time, got involved in pediatric cancer research using artificial intelligence, machine learning to start solving, some real big challenges in that space. Did my postdoc in bioinformatics and molecular biology, so I eventually started, getting more involved in the sort of day-to-day laboratory side of things. All of that was happening at the Children's Cancer Therapy and Development Institute, where I am an assistant member. But really, right now, my main focus is on First Ascent, which is it's bringing functional precision medicine and AI analysis for individualized treatment options into the clinic for patients in need. And I'll start off just by describing, Diana and I are serving as collaborators right now. And, what we're trying to put together is this entire pipeline. The idea of using functional precision medicine to guide individualized treatments. What we're doing together is taking a biopsy sample from a patient's cancer, doing a rapid culture and drug sensitivity testing protocol. All of this using Diana's technology that she's developed over really the past decade. So that's sort of side one of it. Side two is also taking the same tissue and sending it for DNA and RNA sequencing analysis. And where I come in is taking both of those data sets, ingesting them into the AI ML engine that I've also been researching and building over the past decade. The idea there is from the drug testing data and the sequencing data, we can better understand the weaknesses underlying a patient's tumor. Another way of saying that is we take all the drugs that are working, all the drugs that aren't, understanding the mechanisms that are really driving drug sensitivity. So when we try to build combinations around the patient's key mechanistic targets, it becomes a lot easier since it's just a matter of we need to target this gene, we need to target this gene, do so at the same time. And we can find the right drug already available in the clinic that best fits that need. And our goal is to deliver all of this data in under 2 weeks, making this as clinically usable as possible, because that turnaround, that time frame is absolutely critical to meeting the needs of patients. And the really great thing is we've already done a lot of this work in the clinic already. So I'll turn it over to Diana now for talking about all the work she's done in drug sensitivity testing.
Thank you, Noah. So yeah, as Noah mentioned, I mean, the goal of this is to be able to implement this in the clinic. And we have 2 feasibility studies, both in pediatric and adult cancer patients in collaboration with Nicklaus Children's Hospital for Children and in collaboration with Cleveland Clinic, Florida. And the idea is this feasible? Can we recommend treatments in a clinically actionable timeframe? And if these treatments are recommended, how do the patients respond? So here you can see that, this is a 7-year-old girl with rhabdomyosarcoma. It's a metastatic rhabdomyosarcoma and a particularly difficult to treat cancer. This girl has been through multiple treatments. None of them were effective. So we received a piece of her tumor. We confirmed we had the right cells by looking at the different markers of rhabdomyosarcoma. We tested our panel of drugs, and then we ended up testing even multiple drug combinations for the patient. And we were able to deliver this data to the doctor and the molecular tumor board in about 1 week. Move on to the next slide. If you look at her clinical course, you can see Noah. Lost Noah again. Hello?
He may have frozen again.
Yeah.
Deanna, you'll have to improvise until I
yeah, so yeah, let me actually let me try to share my screen again. Let me do that. Sorry about that. Okay.
While you're doing that, I actually am a little bit dangerous because I helped Casey Altman with her hackathon for her, and she had alveolar rhabdomyosarcoma. And this rhabdomyosarcoma I know responds to chemo, but then just about nothing else. So if you find more chemo drugs, you're probably going to kind of delay things, but you're not really going to come up with a durable response. Is that true?
So that's no, I wouldn't say that, just because like in this case, I mean, let's go back one slide, Noah, to look at the drugs that were effective, because yes, we do find chemotherapy drugs that were effective, but we did find the targeted drugs. Noah, can we go back one slide just to? So we did find other drugs. Let me just quickly read. I mean, again, it all depends on the availability of these drugs, right, for the patient. But we did find dasatinib to be effective. We found HDAC inhibitors to be effective, lenalidomide effective, rapamycin, which is mTOR inhibitor. So we did have a couple of options that are targeted drugs. Now, again, one of the challenges about recommending treatments and is really, trying if doctors have access to these drugs. And again, with children, we customize the library of drugs based on what's available in the pharmacy at Nicholas Children's Hospital. And so in this case, if you go back one slide, in this case, the doctors wanted to treat the patient quickly. So they picked chemotherapy drugs, which they thought wouldn't be effective anymore, but they were still sensitive here on the tumor cells. And so when they did that, you can see in the clinical course, you can look at, I mean, if you, before enrolling into our trial, you'll see that her cancer, she only had 2 weeks of progression-free survival. So the treatment wasn't really effective. And then she was enrolled in our clinical trial, and the molecular tumor board recommended treatment based on the drug sensitivity testing. And the tumor really, there was a decrease in tumor size, and we observed a regression in her tumor, and her progression-free survival was 6 months. Unfortunately, the patient passed away because of metastasis in the lungs, and so she had other sites where there was metastasis, but she really did respond to the drug sensitivity. It responded to drug-guided regimen. Another case here that we're going to show is a metastatic osteosarcoma patient, 9-year-old girl. It's a rare bone cancer. Again, this disease is incredibly difficult to treat, especially when it's metastatic. So we received her tumor sample, and again, we did drug testing and sequencing. And what's really beautiful about this is that the results from both the sequencing, the molecular tumor analysis, and the drug sensitivity testing was used to identify a combination that is effective for her cancer. As you can see here, the NTRK fusion, just before you go to the next slide, the NTRK fusion, you have larotrectinib, which is a targeted drug, and that showed up in the molecular tumor analysis. And then when we look at the drug sensitivity testing, the most effective drug here was idarubicin, which was recommended, and also montelukast, which is an allergy medication. So really, this is a beautiful example of how both results from both genomics and the drug sensitivity testing was used in recommending or in her treatment recommendation. And when we looked at her clinical course before enrolling in our trial, her cancer kept growing despite multiple different treatments. And then when she was recommended based on functional precision medicine, she had a complete response, and her progression-free survival is 1.5 years. So if you compare the progression-free survival of the patient based on that was recommended by FPM versus just her previous regimen, you can see that there's an improved survival benefit. So
Diana, just
yes.
a quick question. Was that a drug combination then? Was that 2 drugs?
Yes.
Okay.
So this was the combination of 3 drugs: larotrectinib, idarubicin, and montelukast.
Put a pin in that. We'll come back because drug combinations has been one of our favorite topics.
Yes, and that's what we're doing, right? That's exactly where the machine learning is going to be very important. Slide. So just wanted to show you that we've been able to optimize the drug sensitivity testing on at least 13 different tumor types, and we can do this in liquid leukemias and also in different types of solid tumors, as you can see here in the slide. So, what I've shown you is a few case studies, but this is really the data that we have so far. We just finished, our first feasibility study is just ended, so we're looking into analyzing all the data. But this really, the responses now is not just for 1 or 2 patients. We have a group of patients that we're looking at their best overall responses. And if you compare patients that were guided by our approach versus just their previous regimen or standard of care, you can see that there's a huge improved overall response compared to standard of care and previous regimen. As you can see here, it's really invergent in the outcomes, and the data speaks for itself. So this shows you that it's feasible. We can do this. We can give back data and recommend treatments within an clinically actionable timeframes, and patients that were guided by our approach have improved overall responses compared to their previous regimen and compared to those that went through standard of care.
And
I will move on. Yeah, go ahead.
I'll take over from here. I apologize. I'm going to leave my camera off for a little bit because, again, just getting a lot of internet instability, and I apologize. So as I mentioned previously, my side has been much more focused on the AI and machine learning components of this. And what I've been working on for a little while now is an AI engine that is able to pull in functional drug testing data and sequencing data, integrate it for an individual patient, pulling in as well information about the drugs and what their sort of real world mechanisms are, putting all of this together in order to build out an understanding of the patient's tumor weaknesses and sort of creating a tumor circuit. The idea with the tumor weaknesses is these are the multivariate genetic targets that are really driving drug sensitivity, the main things we really want to hit with the drugs we would give to the patient. And the tumor circuit is sort of the holistic view of what this patient really looks like. So it's, when we put all of these individual weaknesses together, what can we learn about the patient and what can we do to really drive forward individualized combinations? The goal of this has always been to find the right combination for the right patient. And so, this work started in my at the beginning of grad school. So I've been doing this for since about 2012, so about a decade. And what we have done along the way has been to validate this kind of approach out in multiple different cancer types. So what I'm showing here is data from a publication from a couple of years back. This is in a genetically engineered mouse model of alveolar rhabdomyosarcoma. And what we did is we followed the exact same, pipeline that we've been showing so far. We took a tumor tissue from a mouse, did drug testing using 60 agents, some of which were FDA approved, some of which were, sort of in phase 2 or phase 3 studies. Used all of that data plus the genomics data to build a circuit of the patient, finding that this combination of OSI-906 and pictillicib together seemed to have the kind of synergy that we would be targeting, whereas individual drugs may be less effective. Though there's a lot of data here, I'm not showing all of it as in the publication. But the point here is, when we did this analysis, what we found is that the targets of OSI-906 on their own were not critical, nor were the targets of pictillicib. But when we put that combination together, we were finding the kind of synergy in vitro that we were hoping we would see. So when we tried it out in vivo, again, the same sort of approach, the same sort of results replicated. What we see is that the individual drugs are indistinguishable from vehicle, but when we put them together, we see a far more profound effect. We did the same thing in a cancer called epithelioid sarcoma, in this case using a human cancer, which we were able to engraft into PDX mice, so mice engrafted with human cancers. And we did the same sort of approach, drug testing and some sequencing, feeding it into the AI engine to find a combination. One of the really cool things here, though, is because the patient had a relatively large tumor, we were actually able to sort of break it into different subregions of the tumor and do drug testing on each one of them, basically trying to account for intratumor heterogeneity, the property of which, one region of cancer can look a bit different from the other regions of the same tumor. And when we put all of that data together and found this combination of BES-235 and sunitinib, what we see is that that combination outperforms, of course, control, slowing tumor growth by 91%, but also outperforms even the individual drugs alone. And what I'm not saying here is also this is from n equals 3 mice. So even in the minimum statistical sample size, you're still seeing a significant effect. The idea, again, is we can take all of that data from that one patient and build a combination and do so quickly. And finally, what we were able to do, we had, this was an undifferentiated pleomorphic sarcoma. We had the same sort of approach we had in this case, a PDX mouse model line that we were able to access and test on. And so what we did was essentially a full workup of this patient and their associated PDX, doing profiling or DNA profiling to find druggable mutations, doing some drug screening to find possible candidates, even doing some IHC-based profiling, and of course, using the AI approach to put together the sequencing and drug screening data to find what we could find. And what we see from this is that, there are some effective treatments, but the treatment that has the overall lowest tumor volume came from the AI design combination. What we're saying here is that when we put all of the data together in the right way, we can get, we can find better insights to the individual patient and build those individualized combinations. And, sort of finish up, this side of the presentation by talking just a moment about the biomarker side of it. So the same AI that we built to find individualized combinations for patients can be just as easily applied to biomarker discovery for pharma companies. One of the things that we were asked to talk about was, what does the quote unquote business model around this look like? Which is why I'm bringing this up, because if the same AI can be used to work with pharma companies, it becomes a really easy way to monetize a lot of this. Again, it's the exact same workflow, the only difference being a feedback loop that's based on, now we're working with pharma companies and CROs finding the next model for them to test the next PDX, next set of cell lines. And we actually have quite a bit of work in this, but what I'll show briefly is a study we did in PARP inhibitors. So for those who don't know, PARP inhibitors are a relatively new type of drug, a lot of interest in them, and they have sort of a gold standard biomarker, that being mutations in BRCA genes. But from clinical studies, what we've seen is that it works some of the time, but there are some patients without BRCA mutations that will respond, and there are some patients with BRCA mutations that won't. So it's really important to find, better biomarkers that we can add to sort of add to that decision making. And what we did is we took a data set of about 20 ovarian cancer PDX mice, tested with a PARP inhibitor, and used all the genomics data and the response data to find new biomarkers. What we found is that in the biomarker we built, that was really just a combination of a couple genes. Regardless of how you look at the results, whether it's the actual distribution of tumor growth in the PDX mice, or if it's a variety of different test metrics, the biomarker we built using AI outperforms the BRCA-based mutations, BRCA1, 2, or even sort of the BRCA-associated genes. And this holds true even when we apply the same biomarkers to other types of cancer. What we still see is that we are doing significantly better in tumor growth inhibition distribution versus BRCA1, 2, or BRCA-any. And when we look at accuracy, Matthews correlation coefficient, F1 test, or even the ROC curve, we're outperforming the biomarkers that exist today. The point being that when you put the right data together, you can start solving challenges in other fields as well. But building better biomarkers is critical to better assigning drugs to patients in need. And just to really sort of talk for a moment more about the sort of the business model here. There's an entire ecosystem that revolves around getting the right drugs to patients and also building massive data sets. So, the real big go-to-market, so to speak, hurdle that we have right now, that's really just getting CLIA certification for all of this, drug testing, sequencing, and AI analysis. And we are in the process of doing that. Once we have CLIA certification, we can work with any patient in the country from any hospital. But the bigger strategy here is building this massive data set of patient drug response data, matched clinical data, matched sequencing data, and even underneath that, this biobank of all of these tissues that can be used to do new studies. That enables us to work with all of the players in the space, especially pharma companies. If you're looking at this from a business model perspective, what Foundation Medicine did, which has been one of the most successful companies in the space, they built a process that anyone could use, and then they built the data set that pharma companies loved. And we can do the same kind of thing when we put our technologies together. Together we've worked with a variety of different institutions, including Stanford and Johns Hopkins. We've already worked with multiple pharma companies, including Roche, Genentech, and GSK, all on the biomarker side. And with Diana, we have done a lot of clinical work as well with Nicholas Children, the Cleveland Clinic, and we're excited to say we're building relationships with a variety of different centers, OHSU, Mayo Clinic, and so on. And we have published a lot. Here are sort of the top publications for the functional testing side, the AI and ML individualization side, as well as our active and completed clinical trials. And I'll close with an acknowledgement slide. Diana and her team at FIU are incredible. And I have a ragtag group at the Children's Cancer Therapy Development Institute. And like to open it up for questions.
I'll help moderate, and we'll use the hand raising. You see, Saeed is already on the system there. But I'm going to take liberty and ask one question before I turn it over to Saeed, and that is on drug combinations. You mentioned the one example was a case with a drug combination of 3. We have had some concerns of treating physicians that drug combinations might have toxicity issues and drug interactions. So how did you overcome those? What were the arguments that you used? I assume that you were working with a treating physician as a sort of in a collaboration with the insights you were coming up with. How did that all work?
Absolutely, that's a very good question. And one of the the main challenges, right, is to make sure that the combinations of drugs are therapeutically relevant and they're being used before. So based on my experience, what we're doing is we're looking at all the drugs Cmax concentrations, and when we test those drugs, we test those drugs we test the doses based on the Cmax of the drug. That's the first thing we do. And then when it comes to combinations, so far, obviously, doctors are familiar with certain combinations of drugs. So they're more inclined to use those combinations. Now, in cases like this osteosarcoma patient, she didn't have any more options. And so in that case, they ended up treating the drugs in combination, but more in a sequence type of manner. So they gave the targeted drug first, and then they gave her Montelukast, which is an allergy medication, which that's fine. And then they gave her idarubicin in a few cycles. So, it's, again,
Diana, let me just interrupt.
Yeah.
So it wasn't a cocktail all at once. It was sort of staggered. How long were they waiting between the administration of each next drug? So they weren't waiting until, a resistance or something. They were consciously going in, treating like a combination, but in sequence. How long between drugs?
I think this was over, I don't remember right now, but I can go back. We're writing this up. I think it was over 2 months. If I'm not mistaken, I can send you the data. I send you exactly like the regimen and how long they waited, but it was really a matter of giving them all at the same, like mostly over a few weeks, but in sequence, at least in this case. In another case, like the one that I showed you, the previous case, the rhabdomyosarcoma, they gave because they had requested a combination testing of the 3 drugs. And then when we tested the 3 drugs in combination, we found that vincristine alone was more effective alone, a single agent, versus the 2 other drugs. And so they ended up giving vincristine on day 1 and then the 2 other drugs on another day. And so they followed the sequence of the combinations based on the drug sensitivity scores as well. But that was all given at the same time.
Okay. And those physicians, those treating physicians didn't, I guess, by sequencing, was that dealing with some of the toxicity concerns? Because they could see response?
So far I have not really, I think because these patients don't have any more options, so far doctors we're not hesitant or worried about the toxicity. I think they end up looking at how these drugs are given and how they're used to these drugs being given in combination, and they end up deciding based on that.
Okay. Yeah.
So what about dosing?
Yeah.
So yeah. What about, did they change the dosing?
So again, yes, I think if I go back to the trying to remember the vincristine with the other 2 drugs, they did change. No, they didn't change dosing. They gave the doses based on the Cmax, and but they changed the sequence. Okay. They gave vincristine alone because in the drug sensitivity scores, vincristine alone was more effective as a single agent than putting it with temozolomide and irinotecan. So they decided they'll give vincristine alone, and then they gave the 2 other drugs on the second day. And that's the sequence they use.
Okay, so it's standard dose, but spread out differently.
Yeah.
Okay.
And again, I have to emphasize the Cmax is so important. So, again, the Cmax of the drugs, when we test them on the cells, we are using the Cmax. And if we find that the IC50 of the drug is less than the Cmax, that's when we're using the drug. Otherwise, they don't use the drug for treatment.
This is the first I've heard of Cmax, so I'll expose my ignorance. What does Cmax stand for, and what is it?
Cmax is the maximum therapeutically relevant concentration of the drug in the blood. So it's the maximum concentration that the drug can achieve in the blood of patients. And all that information is known, right? Because these are FDA-approved drugs. So we know this is the maximum concentration that the drug can attain in the blood. And so when we do the testing and we find that the drug, is going to kill half of all the tumor cells, but at a concentration that's higher than what can be achievable in the blood, we don't end up using the drug. Yeah. So it has to be within that it has to be within the Cmax concentration and what can be achievable in the blood.
And you just said one more thing, and then I'll turn it over to Said. You just said one more thing. Approved drugs only, and no clinical trials.
So far, we've been testing approved drugs only, but we can also test drugs that are in clinical trials. Absolutely. I mean, there's no reason why we cannot. I mean, I would be very I would actually push for this type of approach because we can guide, patients to clinical trials. So if we have access to these drugs, and which we do, there are many of them are commercially available. We can test them and also guide patients to clinical trials.
Yeah.
Okay.
It's, what we're trying to do is open up as many options as possible. And, again, on the, quote unquote business model side, there's ways to build relationships with pharma companies around this, saying we're more than happy to de-risk some of these patients up front. If, before they enroll in your study, that this patient seems to be responding really well, at least their cells are ex vivo to the drug that you're going to give them, gives them a little bit more confidence. Conversely, if they're seeing no response, it might help say, this is not the right patient for this trial. They need to go somewhere else.
So, and just to underline what you're saying now, I asked you this question before in a side conversation. Would you consider the work you're doing more about functional testing or about AI/ML? And it's a mix of both, is what I'm hearing, because you were just touting the functional testing aspect of it. You're a bit of both.
Very much so. The really cool thing is the functional testing is critical and it has immense power on its own. The AI also is really powerful and it has the ability to take the functional and make it better. It also has the ability to take a lot of functional and make it better for a lot of patients. So, in many ways, we are still exploring all the ways that functional and AI/ML can interplay, especially when you start layering in genomics. So it's hard to say it's not that one is more powerful than the other. It's that the two of them are going to be synergistic and make each other stronger.
Yeah, I believe that too. Sorry, I just want to add that I've been working on functional drug testing for many years, but I always knew that there's something really missing here. We need to know why these drugs work. And with the AI, included AI that integrates the genome, whole exome sequencing and the transcriptomics, not only you're predicting combinations, but you're telling the patient why this is working. And that's where I believe the power of both, integrating both, is what we need to do in cancer treatment.
And let me ask then a question that I'm sort of curious. When you have the functional testing, you have some hypotheses, or maybe you have a standard set. I think you said something like 60 drugs you might test. So your AI/ML based on the DNA sequencing and RNA sequencing might give you some candidates you'd want to try in that direction. But in the other direction, if you do the functional testing and you get different kinds of responses, then the AI can be telling you which ones to prioritize or which ones to combine. So you could use the AI on both sides. So how are you, and you're nodding, so are you doing both?
Right now, I'd say we're really focusing on the AI on sort of the end of it, because that's going to tell us why the drugs are working and help find what really is going to be the best kind of combination for the patient. But yeah, with all the data we have, if we know up front that there's a set of, like if you have, let's say, a PIK3CA mutation, well, that gives a good reason to add a few more PIK3CA targeting drugs onto the screen. It'll help us better understand what that mutation means for that patient, because it might mean that PIK3CA inhibitors will be great, or it might mean that the right one will be really effective, or it might be completely unrelated. It'll help us better understand what that means. So yeah, right now it's the end, but we're going to start layering it into the beginning as well.
And I guess you're working off of fresh tissue, all right, for these. So, and a fresh tissue has like a 24 or 48 hour life. So it's not like you can run one set, hey, come up with a new hypothesis and then run functional testing again without getting a new biopsy. Is that true?
Sorry, I missed the question. I was trying to answer the chat.
Okay, so the question is, you're working off of fresh tissue. Fresh tissue has a half-life of 24 or 48 hours. So it's not like you could run these 40 or 50 or 60 drugs and see what happens, then run your AI or ML and say, oh, I've got a new hypothesis, I've got a new question, I'd like to run these drugs or these combinations. You don't get a second shot because the fresh tissue is no longer fresh after 24 or 48 hours.
So let me answer that. We do get a second shot, and that's what happened in all the cases. Because what we do is we get the fresh sample, we process it, we use what we need for drug testing, and we freeze them down. And then if we need to go back and test combinations, we're throwing those out fresh, and then we're testing within a few days. So we've been able to do that. That's awesome. Actually the approach that we've been doing, and it's very helpful because there's sometimes doctors look at the single agent data, look at all that and say, you know what, these are the combinations I want you to go back and test. And that's what we've done. And it's been very helpful for the doctor.
Okay, so now say, sorry about the interruption.
No problem.
Please ask your question, and then Rebecca.
Very good question. Yeah, actually, my question was related to the concept, the dosage, which you explained. But I'm going to use my time just to share what we found from our own simulation. It's not the lab test. First, we support multiple drugs instead of single drugs. Second, our simulation supports having drugs together instead of one by one, because you give the cells, cancer cells, the chance to adjust itself to the every chemicals you throw at them. And third, using the lowest amount of drugs, not the maximum amount. Because when you increase the concentration, the side effects of drugs can affect the other drugs, right? That's our findings just based on our simulation. Thank you.
I can speak to that for a moment. Yeah, in many ways, I'm in agreement. And actually, some of the other work that I've done, didn't present it here, but has been on showing the difference between sequential combinations or simultaneous combinations, using the AI to find a combination and then sort of testing that ex vivo on a couple of cell models derived from the same type of cancer. Showing the combination can essentially stop tumor regrowth. But at the same time, what we're showing is, just in a practical setting, when you give the drugs at the same time, the effect is that the cancer cells go away and don't come back, which of course is the key end goal. The place where the friction occurs is going to be, giving those two drugs simultaneously may be a little bit much for a patient with less than optimal health. So, we're definitely focused on the right way to give the right combinations. And I guess, at least in my mind, and I imagine Diana, you as well, we're looking towards that simultaneous combination as the right way to do it. But it's, step by step, we have to get there.
Thank you.
Rebecca?
Great presentation. Thank you, Diana, Noah. This is really, really helpful. Just to give you a little context around what I'm going to say, my experience with chemosensitivity actually dates all the way back to 2003, 2004 with some of the original people that were working on chemosensitivity. So, it's been around and evolving. So I say, great work, because I always saw that there was something to it. And so now that we do have genomics and more biomarker testing, I see huge opportunity for this. I have lots of questions for you, but I'll base it on the side of the clinical data. What are your plans for, especially from a laboratory perspective? I do also come from Foundation Medicine, that, collecting some of that clinical data can be very challenging. And as we're combining these drugs and looking at the side effects and dosage and efficacy, what are you planning to do around acquiring some of that clinical data? And I would say even broader in the community setting, because we can run these tests and these studies, I think, a little bit more effectively at the academic level, but we also know that we need to reach patients more broadly in the community setting. So just any commentary on that and collecting clinical data, just to basically clinically validate this test.
Absolutely. Thank you, Rebecca. So the main challenge that we were working on, which we actually just resolved, is getting funding to get this up to a CLIA standards. So we just got state appropriations to set the lab to be CLIA certified, and so we can potentially expand this and really provide this type of service to any, and work with any hospital and any clinical center, because that's important for us, as we know that we need that clinical data back. Nobody has validated that drug sensitivity testing or AI can actually improve survival in patients. That's what's missing. We don't have data in the field showing prospectively that we can potentially use this, and it has clinical utility. We've been working on generating this data, as you saw in the studies that I showed you, but these are small pilot studies with Nichols Children's, with Cleveland Clinic. I've have very strong relationships with the doctors. They know that if they're going to work with me, I need data back. And I think that was key in that was the, what made it successful, in that we got the clinical data back. We're showing that it's feasible. There is clinical utility of this approach. So now the next step is, okay, in a randomized setting, in a large scale clinical trial, does this approach really improve survival benefit in patients? So, and the CLIA certification is going to change, is a game changer here in terms of scaling this up and really getting more data from patients.
Yeah, thank you. That makes sense. It's from a stepwise approach. I would absolutely agree. Obviously, CLIA comes first. And then the other thing, I was pleased to hear you say, just because I don't treat all testing as the same. So, you had mentioned whole exome sequencing and transcriptomics. So do you have a relationship with anybody running that, or how are you going to work with that maybe in your study situation?
Now I do. I mean, Noah can answer, but I'm just going to tell you on the clinical side, we've been doing this. We work with Semaphore. They do whole exome sequencing, and they're really great in terms of a CLIA certified assay. We've also, for children, I've worked with UCSF, and then I'm also with OncoKids. I forgot the name now, but we've been working with a few CLIA certified labs that do this. And it's been great, but again, we're open to other partners who would want to do this with us. We're not planning to do this in-house because it's just really I mean, there's so many good assays out there. But what we're going to do in-house is get all that data and use our AI approach, or AI for analysis.
Just to clarify, so you're outsourcing the DNA sequencing and RNA sequencing. Are you also outsourcing the functional testing, or are you doing that in-house?
No, functional testing is us.
Okay.
Just to be functional clear. Testing is us, absolutely. And the lab that we want to set up for CLIA is my lab at FIU, and First Ascent is also, using that lab.
Yeah.
What about also on the proteomic side of things?
Yeah. I can speak to that side a little bit, as well as the transcriptomics. So, right now there's a lot more focus on the DNA side of precision medicine. So there still is work to be done, even in, as far as I can tell, doing, sort of whole transcriptomics at a CLIA certified level. So we'll take the RNA we can get, but ideally we can eventually get to the point where transcriptome is at a CLIA level and we can access that data routinely. Proteomics, in my view, from what I've seen right now, proteomics isn't fully ready for like high throughput whole proteomics analysis. It's not really ready for prime time. But there will come a point where that kind of data, when it's available, is going to be invaluable, because right now, in some ways, RNA is a proxy for proteomics in its own way. The future I see there is we can integrate proteomics when it's available, and when it becomes readily available, it's going to be really valuable. It'll give us a direct idea of which genes are actively being expressed, what we can actually really reliably drug. And the great thing is, we've already worked with proteomics in the AI side. It is incredibly easy to integrate because it is just quantified data. So when we get to that point, it is going to be folded into the model. We'll do some studies validating that proteomics, really helps refine and strengthen our models. But yeah, we're just waiting for that to be ready to go.
Okay, I have one last question, if it's okay, Brad.
Go.
So you're using a lot of mouse models. So what are your thoughts about the organoid models now?
I just want to add something, and then I'll let Noah. So organoids are great models, but they take weeks to grow. And then in our experience with refractory patients, we don't have a lot of time. And so my approach is really more like a 2D, 3D model. But what's important for us is to be able, we can do the drug testing and deliver data within a week, if not less than that. And that's the only problem with organoids. It just takes time, and you don't get the opportunity to test many drugs. You're limited by the number of drugs, which in our approach, we can test hundreds of drugs within a few days. So I believe that our approach is is better in terms of delivering results in a clinically actionable timeframe.
Yeah. So what's critical here is to start getting the physicians to ordering sequencing upfront. That's the
Yes.
challenge.
Yes,
So
and
that
that
we're
takes
not waiting
weeks.
for another
Uh,
analysis of weeks. Yeah.
I know, and that's one of the challenges we had when we were, going through all the feasibility studies. Is like, sending these cells out, sending this, getting the data back from sequencing takes weeks. But it's still useful. It's still helpful. I think with the AI, we can, NOAA has been able to take the drug sensitivity data and then slowly, get in. When we get the sequencing, we can always modify the combinations based on the sequencing data as well, as we can recommend treatment, the patient's on treatment, and then we can go back and slightly modify the treatment based on that data.
Yeah.
So we've been able to manage
Great.
doing that.
So I have lots more questions, so maybe I can meet with you guys at another time. So I don't want to hog the floor here, but thank you
Thank
so
you.
much.
Yeah, we'd love to meet with you again. If it's okay, I have a stream of questions from Rick that I'd like to address real quick.
First, let me say hi.
Hi, Rick.
Electrical engineers love electrical engineers. You know that, Noah. So I'm an electrical engineer, and
Yep.
I think you're great. Sorry I'm late. I had a rough night. I'm stage 4 prostate cancer. Just had 11th round of chemo, and it's still got a little grip on me. So sorry I was late. Yeah, you can tell I'm asking AI kind of detailed questions. So anything you can shed light on with the understanding that I dig you guys, please share.
Yeah, absolutely. So I'll just sort of scroll back. All right, so NanoString might disagree. At least what I understand of NanoString right now is they can do some targeted proteomics, and that can be really valuable. But what I'm thinking about is sort of large scale, all of the genes that can be expressed. What is the, sort of protein activity level of all of those genes? In my view, that's when things are going to really transform, because we will be able to deeply understand, first of all, where the activity is, and also how does the activity change in the presence of specific drugs. I think we don't really have a high throughput way of doing that yet. I think, there's like, if you're looking at specific proteins for specific treatment responses, there's a lot of potential, a lot of power there. But as an engineer, I'm always looking at like the biggest data set I can get. Who interprets the outsourced sequencing? That's me. I've done so much bioinformatics work. Back at CCTDI, I actually built the pipelines from scratch for all of the sequencing analysis, and we're publishing on it. So, that part that's kind of my domain.
So you understand cancer signaling inside the tumor cell pretty well?
Well enough to be able to do what needs to be done. It just at a personal level where I don't always do well is I can't, somebody can't throw 30 genes at me and say, tell me what's going on here. I need to look a lot of stuff up, but I know the way to put all the data together so that we can, is what I would
Do you
say.
use QIAGEN tools?
QIAGEN,
Or that level of,
kind of. A lot of it is genomics databases. And because, there's a heavy focus on the functional side, a lot of it is also drug target interaction databases as well. But, a lot of ontology tools, GSEA, pathway analysis kinds of tools. Yeah.
Yeah. Okay. On whole transcriptomics, how do you normalize your data?
It really heavily depends on the study. For the kinds of work we've been doing, the biomarker development, that side of it, the way we normalize is using, it's GETMM is what I've been primarily using. It sort of does pool normalization across a cohort of samples. So, sort of saying your study in a way is self-contained. What is the study telling you? And the nice thing is when I've actually sort of taken that normalized set and expanded out to include more, what I see is that the actual values post-normalization don't really shift that much. So once you hit a certain number of samples, let's say 20 or 25, adding the 26th doesn't really significantly change the expression level. As for the individual patient, What we've been doing, what I've done in the past has been using a sort of a pool of other samples, both normal and cancerous, from a variety of different databases like GTEX or some other similar databases.
But GTEx is normally captured with a different RNA capture technology, isn't it?
Yeah, in that sense, it's not ideal. With the RNA-seq normalization, I would say it's kind of best we can do. And this is a debate I've had with I haven't talked about him at all, but Charles in the middle of, my picture. We will routinely have debates about what is true normal in RNA-seq because, let's say because we focus on sarcoma, is true normal damaged tissue that's actively regrowing but not cancerous. It is really difficult to establish a normal in RNA. So, there's sort of ways we go about that. I would say what we're really looking for is trends that are overlapping between the functional response data and the sequencing data. So we're not trying to make decisions from sequencing alone. We're putting it all together to really glean insight.
Perfect.
Yeah.
Love that. It's been a goal of mine as well. Curious, when you say AI, I know you.
Yeah.
Any architectures you're talking about? Any the type of inputs?
Yeah.
Because right now, I think you guys are great. I want to learn more. Maybe do me. But right now I see this hand wave of we use AI, don't ask beyond the curtain of the great and powerful Oz here. And so just any clues would be
Absolutely.
welcome.
Yeah, first of all, completely agree. There are so many groups that say we're doing AI and it's just, we're looking up stuff in an Excel table. So what I will say about what I'm doing is it's mostly based on what's called probabilistic Boolean modeling, which actually puts it more towards sort of, we really say it's more causal machine learning than anything, but right now AI is the buzzword, so that's the word I default to. But the idea with probabilistic Boolean modeling is you're basically taking, you have your response feature, in this case, that's drug sensitivity. You have your feature set, which in this case is going to be a variety of different things, including drug targets, sequencing or RNA data, DNA data, copy number variation data, whatever kind of data you can really build from your models, whether it's an individual patient or, cell lines, PDX models, et cetera. It's basically taking all of that data, finding ways to sort of normalize it into, Boolean variables, true, false, putting all of that together to start finding relations between all of those Boolean features and your response feature, drug response. And then sort of, taking those top relationships, layering on additional data, that's sort of publicly available or private data to understand what all of those features are really telling you. So, what that really means is when we find those features that are really strongly correlated with drug response, either to a specific drug or all their drug response for a patient, and then put together, a lot of additional data about what these targets really are, it starts creating that story, that understanding, and gives us insight as to, where to take a drug or what to give that patient. So that's my, shortish answer. If you want to dive through all the publications, please do so. I'm happy to talk through them with you.
I have a related question, Noah, which is, let's suppose it's a black box and you and Rick just had a conversation when you geeked out and, really went into the, the details. But I'm now, again, a treating physician, and your AI has spit out that these are the 3 drugs that I want to run in a combination. Why should I believe you? So it's a question of, it's almost like a question of trust in the predictive accuracy of whatever you've built. And if you haven't compared it to a randomized clinical trial, how do I know that your predictive model is worth anything?
Absolutely. So the first answer is, the work we've been doing to validate it, at the very least, shows the potential that when you design a combination this way, you do see a trend towards synergy and sort of a better outcome from your study. There is clinical work to be done in this, absolutely, and that is a threshold we'll overcome. We have our plan to do that. But first, the reason that it's worth exploring is when we've built combinations this way for individual pseudo patients, we see the outcome we were hoping for. The combination does better than the single drugs alone. Second, the reason that it's worth trusting, at the very least to try out, is because, you know, there's a lot of black box AI systems that'll just say, here's your answer, don't ask why. We're very specifically focusing on the reasons why, because we're spitting out a combination, yes, but the reason for that combination is this gene's important, this gene's important, and this gene's important. Here's all the data that's pointing towards these genes being important. So, you know, in that sense, it is what you could call an explainable AI. The rationale behind your decision is actually pretty clear. So it becomes a matter of, if the physician wants to dive in that deep, do these genetic targets make sense from all the data we're seeing? You know, these drugs are working pointing towards this, these drugs are working pointing towards this. These genes also happen to be mutated. This one also happens to be really highly overexpressed at the RNA level. It actually makes sense. You know, that's sort of the rationale.
Have you gotten it wrong? I mean, there's a lot of steps in this process. There's a lot of points of failure. And yeah.
Yes, there is.
Right. It's very
Yeah.
complex. And
Yeah.
this whole notion of a black box is a real issue, because decisions are being made based upon this data. I mean, you know, we've looked at my data many different ways, you know, transcriptomic information, et cetera. And we have gone back and we have questioned whether or not what we're seeing is actually accurate. And so, have you run into an instance where you actually got it wrong?
On the AI side, not yet. You know, I guess I would say at every point there is going to be a situation where your outcomes are not what you expect it to be, and I'm fully aware that is going to happen. We haven't hit that yet. I know we will, but I think what we really are going to be able to show is, you know, 70-80% of the time the combination I recommend is going to be helpful, and it might be more helpful than any single drug alone. It may not be the perfect answer, but it's going to point you in a solid right direction. You know, that's the answer there. On the functional side, I leave that answer to Diana.
So again, you know, the data we're showing you is really would try to answer your question is, you know, when these patients were guided by functional drug testing, how did they respond to treatment that was guided? So, and we're seeing so far that, you know, they are responding, which tells you that, you know, our assay has clinical utility. I think that's the data. But again, you know, to really be able to tell the doctor, okay, this is the randomized clinical trial. These are the patients, you know, that were recommended, that were guided by this approach versus patients that didn't. This is the data. We're not there yet, and that's the goal. And I think when we get that data, we'll be able to resolve many of the, you know, unanswered questions. But at least in our experiences, when we do the drug testing, what I've seen that's really helpful is we know drugs, we can tell which drugs are not going to work. I think that also is an important aspect because
Huge.
of the idea right now, the trial and error that physicians have to sometimes go through.
Yeah.
I think that is very critically important. And I've noticed that this has been very helpful for physicians, that they can tell, okay, these drugs don't work. Let me look into other drugs. I think that's the functional drug testing has been very helpful in that aspect.